Welcome everyone that's been joining. So we're going to get started in just a minute or so. We've got a lot of people still in the lobby joining. If you could just please take a look at your microphone and camera and please have them off. Thank you so much, and we appreciate you joining here today. We have quite a few people joining still. So again, I'll just repeat myself. We're going to get started in just a minute. Please just make sure that your microphone is on mute and your camera is off. Thank you so much. All right, a whole bunch of people just joined. So again, I'll just repeat myself. If you could just make sure that your microphone is off, as well as your camera. We're going to give you the opportunity here to put any questions in the chat. But we're going to wait just another minute or so because lots of people are still joining this morning. So thank you so much. All right, everyone that's joining. So just a few things. We're going to get started in just a minute or two. We still have quite a few people joining. Please just make sure that your line is on mute, and your camera is off. So we appreciate it. All right, we still have a steady stream of people joining, so I think it's best to wait a minute or two, and then at that point we will get started, just to keep everybody on schedule. All Okay, I think we're going to go ahead and get started. There's still quite a few people joining, but let's go ahead and get started so that we can stay on board. This is going to be a recorded webinar, and we will have it for future viewing, which is super great. So, and thanks everyone who's making comments in the chat. We're really excited that you're here with us for the next 2 hours. And so, next slide, please. We're going to be talking about mosquito identification. And so, this is supported by the City of New Orleans. It's our speakers here today, our taxonomists, our mosquito professionals that are hosting this webinar, but it's done in conjunction and supported by the Gulf South Vector. I really recommend that everybody go to vectoroutreach.org. We'll put it in the link for you and sign up to get notification about trainings. We have lots of training that's happening in person as well as online. This is a Centers for Disease and Control and Prevention project. We're entering our 4th year, and the goal of it is to strengthen the capacity of prevention and as well as control of vector-borne diseases. So we cover mosquitoes here today, but we also work with fleas and ticks and other arthropods as well that are important vectors. So we have quite a few amazing webinar series that either have been done or are being launched. So I think it'll be beneficial for everyone. So the best way really to know what's going on is vectoroutreach.org. The other group that's supporting is Mosquito Beacons. And so Mosquito Beacons is a work group that supports and really educates on invasive mosquito species. So our 3rd speaker in this series, Mike Riles, is going to be talking about many of those invasives species that we need to worry about in the Gulf South. Today's session is May 15th, that's today. Our next session is going to be May 22nd, and then we'll close it out on May 29th. So if you've already registered, please know that we'll get the link again for that. And just when you type in, just really be very careful so that there aren't any typos so that you're able to get the information. All right, so let's do some rules of engagement. That'll be the next slide. We have some webinar procedures. Please keep your line on mute just so that everybody on the call can hear. We have quite a big group of people attending today. And please use your chat box. We're going to be monitoring the chat box throughout the webinar so that we can save up your questions. You can also use email education@nola.gov. So let's say we just don't run out of time and we just don't get to your questions. Please email them to education@nola.gov. And what we will do is to get it to our speakers and make sure that you get an answer. This webinar is going to be posted on our YouTube channel. So we just need to prepare it before it gets done. And we are going to be putting some poll questions throughout this presentation. So please stay engaged and answer those poll questions. It has to be in a timely fashion. We're going to be offering a certificate of completion. We have quite a few people from outside the country. And so, we'll be offering some certificates of completion. And there are Florida, as well as Louisiana, has continuing education credits approved. And you must attend the 2-hour session and complete all the poll questions in order to receive the certification for those CEs and receive certificate of completion. Next slide, next one. And if you need a certificate of completion, please email education@nola.gov, and we need to have some information included. Alex, next slide, our next button. We need your full name, your title. So you might be an engineer or have a PhD or a master's in public health, whatever it may be. Just make sure that you include that so we can put that in your title. And we need your email address, which you'll be sending it, so it should be there, but just make sure we have that information so that we can get it to you in a timely fashion. So with that being said, I'd like to introduce our speakers today. These 3 gentlemen work for the City of New Orleans. They have amazing taxonomy skills. So we've got Alex Pavlakis, Eric Guidry, and James Beck. They've prepared a really detailed, presentation for you. All of them have tremendous experience in mosquito abatement, mosquito control, surveillance, and are amazing taxonomists. So they put the data or the information here today for you that I think everyone's going to really get a lot of benefit out of it, starting in the beginning for taxonomy and what you need to do. And they're going to be getting more and more detail as a presentation goes on. So with that, I'm going to kick it over to Alex, and he's going to take it away here. And of course, he's going to tell you what we're talking about. So everyone, thank you again for attending, and we'll be monitoring the chat for you. And if you have any questions, please be sure to put it in there. All right, thanks so much.
Good morning, everyone. So I'm here with James and Eric. So we're so excited to be able to talk with you guys about mosquito identification. We know that there's various different levels of mosquito ID here represented in this call. So for today's session, we're going to start at the basics, going over adult mosquito anatomy and morphology, where we'll go over the mosquito basic characteristics, how you should orientate your mosquito, and how to understand that language of orientating your mosquito. Going over some anatomy and morphology, and then looking at the differences between males and female mosquitoes. Then we'll move on to genus level identification. So we'll kind of go over dichotomous keys and go over kind of a example of with live pictures of specimens and have a little bit of a genus level review. And then for our specific genera that we're going to go over today, we're going to go over Aedes species in the Gulf South and Anopheles species in the Gulf South. And with both of those, we're going to be looking at species that are commonly misidentified with each other and looking at a couple different scaling patterns and other morphological features that we can look at to speciate them and understand them. So with that, this course is, we offer this course in person as well. So we have our Mosquito Academy coming up in September 22nd through the 24th. The first 2 days are a really great hands-on training following the IPM pillars for mosquito control. And then on our third day, we have a really amazing hands-on identification course. So this course that we're showing today on the webinar, we get to do in person with everybody gets live samples that they get to play around with themselves and really, get a lot of experience seeing them. So if you like this talk today, please think about coming and joining us in September 22nd through the 24th, and you can register using the QR code shown here, and we'll have it at the end of the presentation as well. So first, we're going to start off with adult mosquito anatomy and morphology. So I'm going to hand this over to Eric, but we're going to all kind of be talking throughout this to keep, this more fun and open.
All right. So everybody on this call probably has a different level of education when it comes to insects or mosquito identification, but we have to start at the basics. So I'm sorry if this is an overview of what you've already known. But basic characteristics of all insects, of course, they have the three body segments, head, thorax, and abdomen. They can have two sets of eyes, either two compound eyes or ocelli, which is 0 to 3 simple eyes. They also have the three pairs of jointed legs hooked to the abdomen. So as you can see in this picture, so my laser pointer's on, we can all see that, and we probably all know that. But to go into a little more detail, all mosquitoes are in the order Diptera, Diptera meaning die, meaning two, ptera meaning wings. They have these structures in the back called halteres, which is common or it's on all flies, which are kind of used as gyroscopes or used for balance. So it's pretty much just a modified hind wing on flies. We know that all mosquitoes are in the family Culicidae. Just throw that out. I don't think I had that in there, but there you go. That's what they are. If we look at the side view right here, we can easily see the head. The middle part is the thorax, which comes into play with identification certain areas, and we're going to go into more detail about that. And then we have the abdomen right there. On the right, you can see it from the dorsal view. So, oh, here it is. So the family Culicidae, well, what makes them Culicidae? Well, they have that proboscis, which is their piercing, sucking mouth parts. Both males and female mosquitoes feed on nectar, but it's only the females that will have to take a blood meal, which is useful for egg production. With their wings right here, we know that their wings are the same length as their abdomen, which is important when you're trying to distinguish them for other things, say like non-biting midges, where their wings will be shorter than the abdomen. They have these scalings on the wings. Certain families or certain genera will that scaling is really important more than others. So whether they have dark patches here or light patches here, what color the scales are on the wings is really important as well, especially when we're talking about things, the Anopheles. Even the size of the scales are important as well, and we'll go into more detail about that as we progress. So, a couple terms that we're going to use throughout this talk, things like dorsal. So, as you can see, looking at the backside, posterior meaning towards the rear end, anterior meaning towards the head, and then ventral, which is coming up from underneath, as you can see right here. And if we mention lateral, it would be on either side of the mosquito. So here's just another overview of the same thing that I just said, just in different views. If we're talking about wings and we're talking about anterior, it would be towards the top. If we're talking posterior, it would be towards the bottom. And as I mentioned before, dorsal and ventral, top of the wing or bottom of the wing. So when we talk about legs, there are a few other things that we're going to talk about. Of course, the first segment of the leg would be the femur. This middle section, which is really important as well, is the tibia. And then we have our tarsomeres, which are the last few segments, as you can see, 1, 2, 3, 4, 5 right there. And then on the tip of those tarsomeres, they have tarsal claws, which is used for gripping and things like that. If I say proximal, it would be the part closest to the body, whereas if I said distal, it would be the part furthest from the body or the unattached part. Another part, as we already said.
I think you went back.
Did I go back?
Yeah.
Oh, I went back. Sorry. There we go. My bad. I just dotan that. So, as we said, compound eyes right here are the main eyes on the head. Antennae are important, especially when we're talking about males and females. I'll show you that in a little bit. The clypeus, which is it's usually referred to as commonly the lip. I don't know if that's really true, but that's this area right here for identification that will come in play quite a bit. Palpi are really important. That could be this segment right here, whereas in males it'll be longer, females it'll be shorter, unless we're talking about the Anopheles, and we'll talk about that later today. And the part that everybody's concerned with is the proboscis, or that feeding mouth part right there. The proboscis is composed of two mandibles. Females use that mouth part to pierce and suck blood from the host, and that's why we're all concerned about the culicids. Clypeus, right? The clypeus right there, as I mentioned before, is the broad plate on the front of the head. This will become into importance when we're talking about scaling on certain mosquitoes, whether they have what color scales, how many scales, and things like that. When we talk about the antennae, of course, there's different parts of that as well. This doesn't really come into play too much, except for one genera that I can think of, or one genus that I can think of. In other insects, the antennae are really important, like say we're talking about ants or things like that. With mosquitoes, it's not crazy important for identification, but just so you know, this is what they have. Of course, we have the scape right here, the pedicel, which will come in handy when we're talking about identification of things like Mansonia. Then we have the flagellum, which is composed of these individual segments called the flagellomeres. Males from females, really easy to do. There's a couple different ways to do it, but if you look at the antennae, this is the easiest way to do it. As you can see on your left, the males will have these bushy antennae, which serve for detecting females around. And if we look at the females, they're more thread-like, and they don't have all that CD coming off of them. There is another way to tell them apart, which is the palps, as I mentioned before. If we look at the males, which in the red rectangle right there, the males are going to have their palpi are going to be equal length to the proboscis, whereas if we look at the female on your right, they're going to be shorter than the proboscis. As there's always an exception to the rule, as we talk about always in the insect world, Anopheles females will have the palpi as long as they're proboscis, but we'll go into that later in this talk. The scutum is really important. It's one of the first things we look at when we're doing identification of mosquitoes. So the scutum is pretty much this dorsal side of the thorax right here. And there's all different types of patterns on there. They can have stripes. They can have different colors, seedy. Some of them are fine stripes. Some are broad stripes. We're going to get into all of that throughout this talk today. Two wings right there. We know we already talked about that. Two halteres. The three pairs of legs, I didn't mention this before, but there's the foreleg, the mid leg, and the hind leg. Obviously, that's pretty telling right there. Forelegs are the first one, mid legs are the middle ones, and the hind legs, of course, are going to be the last ones. If we look at the side of the thorax, there's certain structures on there, different spiracles right here used for breathing. And those placements or where the scales around those are, or seedy, are very important for identification. And that's going to come into more play next Friday when we talk about Culex, but not so much today. If we break it down, there's different parts of the thorax. You can see on the left that this is an actual Aedes mosquito. And if you look at the drawing on the right, it looks more detailed. So we kind of wanted to show you where these different sections are, because if you go through dichotomous keys, they're going to talk about things like, I don't know, the pre-sporacula era, or where the coxa, or all of these things. So this is just kind of a breakdown of where these different sections are. It takes a little while to get used to this, to see where you're looking, because obviously your specimens that you bring in from the field are not going to have lines drawn on them. I mean, you can draw lines on them if you want, but we don't usually do that. So this will be shown throughout the talk as well, to where we're looking. Wing venation, all insects have these veins on their wings if they have wings. This is pretty important, talking about Anopheles especially. So we abbreviate things like C, which is that top wing vein right there, which is the costa. Not so much well, it's kind of important when we're doing identification for certain species. So we just kind of want to know what these are. The subcostal vein, which is the one below it. Radial vein, which helps it rotate, is the one below that. Then we have the medial vein or central vein right there. That's really important. Oh, Christine left.
See you.
Bye. Anyway, so the cubital vein, which is CU right here. And then, of course, the anal vein is the last one.
Yeah, remember that CU vein.
The CU vein is going to be very important later today, in about an hour and a half, I'm sure. So more about the legs. Sorry if we're boring you.
Segments.
What's that?
Segments.
Segments. So
So Eric did talk about, the morphology a little bit of the legs, but the main thing to remember with the legs right here is that these tarsomers are segments, and a lot of the banding patterns that we're looking at are repeating on these tarsomers, and where that banding is occurring on that tarsomer is extremely important throughout, especially our Aedes identification today, but throughout all identification.
Okay. So the abdomen gets important as well, whether we're looking at the dorsal side, and as Alex just mentioned before, where these bands or scaling is present on the abdomen, it will be important eventually. The abdomen is comprised of 10 segments. You usually can only see 1 through 8. Segments 9 and 10 are either reduced or modified, used for reproduction. So here we can see the 8 segments on the abdomen, 1, 2, 3, 4, 5, 6, 7, 8. As you can see, 9 and 10 would be not exposed in this specimen right here. So each segment is made up of a tergite, which is on the top of the abdomen, or a sternite, which is on the bottom. And we use both of these sides to identify certain species of mosquito. This slide is pretty important when we talk about things of scaling, when we're talking about basal or apical banding. So this gets tricky for a lot of people to understand what it is. So if we just take this segment right here out, and if I want to say, does this mosquito have basal banding, which would be right here, it's at the front of that actual abdominal segment right there. Whereas if I say, is it basal, or I'm sorry, apical, it would be here. So the way I usually remember it, B for basal is before this segment, and then A for apical would be after. So before and after to remember where those banding segments are. And I believe we're going to have a picture of this coming up soon. The other way to identify males and females mosquitoes, besides using the antennae, is we can look at the last few segments of the mosquito. So these little claspers right here are going to tell you whether it's a male or a female. And as we can see, here's an actual picture. So the one on the left, which is a female Culex quickofasciatus in the dorsal view, will not have those claspers, whereas we look at this Psorophora on this side, it has this little process right here, which is used for mating and whatnot. So if you just have an abdomen of a mosquito and you don't know whether it's male or female, which we count all of ours, so we want to know if we have males or females, we can just look at the abdomen and be able to tell that, even if the front side's missing. So just to get to the genus level, one of the first things they will ask people using dichotomous keys, well, what does the abdomen look like? Does it have this blunt tip right here, as in this Culex specimen right here? As you can see, it's not pointed at all. Whereas when we look at this Aedes vexans right here, it will have a pointed abdomen. Then there's this other one, which we'll get to next Friday, that has a blunt abdomen as well, but there's certain processes on this species, not the whole genera, where you look for that to see what we're dealing with. But I'm not going to go into too much of that today at all. So, first quiz, that was pretty basic, pretty easy. Is it a mosquito or is it not a mosquito? So, it's labeled A, B, C, and D, and you guys can go ahead and tell me which one is the mosquito, I guess, in comments.
Yeah, a little pop-up should show up for you guys. So we're gonna have two slides. The next two slides are gonna be a quiz, so we'll give you like 30 seconds to get your answer in, A, B, C, or D, which is a mosquito. So hopefully that pop-up came up for you guys.
Bonus points for whoever can identify
did.
C to species.
Awesome.
Gotcha. And Zach Lemon is excluded.
So we're gonna do the answer for this one
Is everybody
right now. So the answer for this one is going to be D. So if you guys.
So we always get calls saying that we have mosquitoes through the public around here. And so A is not a mosquito. I mean, all of these are flies, right? They have the two wings, they have the halteres. A is actually a crane fly, which are common here in southeastern Louisiana. People have referred to these as mosquito eaters, and they think they eat mosquitoes, but actually, as adults, they don't even have working mouth parts, so they don't feed at all as adults. So these are not biting people. The most common one we get, especially now or this time of year, are these non-biting midges. We can tell this is not a mosquito. If we look at the mouth parts, it does not have that proboscis right there. And if we look at the wings, they're shorter than the abdomen, whereas mosquitoes on this side will be pretty much close to the length of the abdomen. This one, D, the answer, this is actually a non-blood feeding mosquito, but we can see those piercing, sucking mouth parts on this one. This is Toxorhynchites. And of course, if anyone answered C, well, that's not the answer. So that's actually a species of robber fly. It does have these piercing mouth parts, but it's obviously not a mosquito.
Yeah, they're using neurotoxin.
So we'll give you guys another chance. So again, another quiz, A, B, C, or D. Which one's a mosquito? And then we will talk again a little bit about it. So we'll give you 30 seconds.
Okay, there's actually a whole bunch of people in there. This is Claudia. We're just monitoring the chat that their poll is not working. So we're not 100% sure why not. I think quite a few of you are not having issues. So just bear with us. Don't stress about it. But if you can see the poll, just please complete it. Thanks.
If it's not working, you automatically get an A.
It sounds like a good plan.
Awesome.
Never had that in effect when I was in high school. Had it straight A's.
We win.
Gone.
All right. So as we mentioned before, A is that tipulid or crane fly that we were talking about. C is that non-biting midge that I just mentioned. And this one, the B is the answer. Of course, it's not Toxorhynchites because you don't see that curved proboscis, but this is a blood-feeding mosquito right here.
So again, the important things that make mosquitoes a mosquito, right, is that A, that proboscis, right, that mouth, that's multiple different mouth parts that make up the proboscis. And then, as Eric said, when you're looking at your wings, that there are wing scales present and that they are as long as the abdomen. Cool. All right, so our next section, we're going to go into genus identification. So just some notes for genus level ID, right? If you're new to your identification, career, getting to the genus level is a really amazing start. You can get a lot of information about like your disease risk, mosquito habitats, and more just from getting to this level alone. And so really for every single person that's in here, you really want to figure out what's the purpose of your ID program. If you don't really care about that species level data, that genus level may be good enough for you to get the answers that you need. But if you need to get to that species level, then again, this is a great first start to get comfortable with, and then you can start getting into that later on, the species level later on. What we always want to reiterate here is to never force an ID, right? Depending on the traps that you use, the conditions in those traps, et cetera, like if it's raining or you have overcrowded with other bycatch or other mosquitoes, you might have various different qualities, qualities in your samples. So never force an ID. Getting to the genus level is fine enough in some cases. And you want to familiarize yourself with your bycatch in your area because that can really help you in that like sorting stage when you have, here's a great picture of one of our traps that we put out in New Orleans East, had over what?
This one had 29,000.
29,000 mosquitoes in one night, right? So familiarizing yourself with bycatch helps get rid of a lot of the mass that's in this plate, but also, being able to just quickly get to that genus level can really help us understand. And then later on, we can get to that species level. So we can understand where these mosquitoes are coming from, maybe what we need to target to do treatments so that we can control this outbreak of population.
I don't think you can accentuate the don't force an ID point enough. And this holds true for not just mosquitoes, but a lot of things in biology. You're really not gaining anything by saying, well, this is probably an Aedes infirmatus because we have 28 others in the trap. If you can't get to species, there's nothing to be embarrassed about there because you're not going to gain anything by just making something up. We're going to say that probably 42 times during this talk.
That was
Oh.
I said it 3 times just now.
So if you've never used a dichotomous key before, this is a really great tool for identifying objects or organisms by making a series of choices based on contrasting characteristics. So many dichotomous keys will have descriptions about what you're looking at, and then also usually like pictures or diagrams as well. but each dichotomous key is different. There's many different keys that you can choose from to use, but I would say we say it's obviously best to use one that specifies the mosquitoes found in your region. But this little asterisk here is that mosquitoes are constantly moving. We have new species that can come into a community, and so if your dichotomous key doesn't include a specific species, you may be misidentifying. So keep your knowledge base up, look at other dichotomous keys, go to events like these to kind of see mosquitoes that may not currently be in your region or your area, but they may sometime come there and establish themselves. With dichotomous keys, there's keys for various different life stages. So you can have keys for adult female genera. You can have it for specific for specific genuses, and you can also have it for that larval stage as well. And what we're going to go over right now is that there's a lot of these terms that can be really subjective, which is really difficult for people that are first starting out to know if something is broad or narrow or curved or not curved. So this next section right here, we're going to go through genus level dichotomous key with pictures to really describe the differences in these subjective terms.
Another term we'll use on a regular basis today would be variability, because a lot of these species are variable and it's not cut and dry. And a lot of morphological features can carry over to very similar, like sister type species.
Cool. So throughout this, just imagine, you have a sample in front of you. And so you can go throughout this in any order. But we're just going to follow the order that was in the dichotomous keys that we use here. So the first feature that you can look at is your proboscis. So the choice is going to be between whether the proboscis is curved downward or not curved. So if we look at our two pictures here, on the picture on the right, you can see a slight curve, but when they're talking about curved downwards, they're really meaning this pretty obvious, remarkable curve in that proboscis. So it looks kind of like an elephant trunk. So if your sample does have that curved proboscis, you're going to immediately think of Toxorhynchites, right? So you can kind of be just done. You know that you have that Toxorhynchites species in your sample. Whereas if it's not curved, you can then move to the next step. So then our next step is to look at our palpi. So we're looking at the length of the palpi. So are the palpi as long as the proboscis, like on the picture on the left right here, or the palpi shorter than the proboscis? And this is a pretty obvious or a significant length difference when we're looking at our palpi. We're not seeing ones that are kind of midway through, right? It's either the same length as the proboscis or much shorter. And then
that, as I mentioned before, this is where the antennae come in handy, because we want to know if we're dealing with a male mosquito or a female mosquito, because as I mentioned before, all males will have those palpi the same length as the proboscis or longer. So this is where it's really important to know whether you're dealing with a male or a female mosquito.
Exactly. So remember that we have those three different ways that we can look at our specimen to see if they're male or female, right? We're looking at those antennae, if they're bushy, the palpi, if they're as long as the proboscis, and typically will be bushier, but depending on the sample, the quality of your sample. And then we're looking at that last abdominal segment to see if we have those cerci visible or those claspers versus not having them. So remember, first get to that level, male or female, then you can start going through these steps. So if you have palpi that are as long as the proboscis, immediately you're going to think, right, I got an Anopheles in my hands. And if they're shorter than the proboscis, we can move to the next step. So the next thing to look at is these thoracic scales or scale colors. Now, coloration is something that we'll mention a lot, and it's sometimes used a lot in mosquito identification, but colors can vary a lot. But in this case, when we're looking if there's these shiny blue scales present on the thorax, scutum, and wings, or there's no blue scales present, this is going to be consistent for this genus level determination. So if you have these shiny blue scales throughout the body, whether it's on the thorax, the scutum, the wings, the abdomen, you're going to be thinking, you're going to immediately think of Uranotaenia. Whereas if you have no blue scales present, then you can move to the next step. So next, this is a very difficult one, but we have some pictures later that will help see this a little bit better. But when we are looking at our wings, if we look at the base of that subcostal vein, will that sample have a row of CT present or absent? So when you are looking at like a gross specimen, you're looking at from afar, these can be pretty evident. We like to call them armpit hairs because they're pretty visible just from the onset. Really looks like your mosquito is just growing it out for the summer. Being a hippie. But you're gonna look at that base of that subcostal wing right near where it's attached to the thorax. So if those row of CT are present, you're gonna be looking at your Culecida genus. Whereas if these are absent, we can move to the next step. So staying on the wings, now we're going to be looking at the scales that are on these wings, right? So when you're looking at dichotomous key, it's going to say, are the wing scales broad or narrow? So when we're talking about it, if we look at these pictures here, on the left-hand side, these are our broad wing scales. So they kind of look like oak leaves. They're going to give these mosquitoes pretty like salt and peppery kind of looks because these typically are mixed with dark and white scales when you see them. But they're very, very broad, and if you can focus in with your microscope at your highest what's it called, magnification, you'll be able to really see that shape of like a oak leaf. Whereas when we're looking at our narrow wing scales, I like to think of it as pine needles. So it's a lot less remarkable in terms of like coloration and how obvious it is. But if you really focus in right here, you'll see this pine needle-like structure here.
So dealing with the broadwing scales as well, this will be next Friday. The type of broadwing scale will come into play as well. So what that tip of the scale looks like will come into play, as I mentioned before.
Yeah,
I do like the oak and the pine leaf comparison there for the scale shapes. But for those that are not into botany, another thing I like to use is if you're looking at the wing scales on the left, kind of like the shape of jasmine rice. And then the wing scales on the right is long grain rice. So it depends on what you want to put in the gumbo.
Yeah. Depends how hungry you are.
Quite
We're always hungry.
So yeah, if you have broad wing scales, immediately you're going to be thinking about your Mansonia and Coquetidia. And if you're having your narrow wing scales, you can move to your next step. So let's say our sample has all the things that we said before to move on. And now we're going to start looking at our abdomen shape. So Eric talked about this a little bit, but mosquitoes are going to have a lot of different ways that their abdomen kind of looks with different morphological features on it. But the first thing we'll look at is just the shape at the end of those last segments. So on the one on the left, this is going to be our bluntly rounded abdomen right here. On the right, we have our pointed abdomen. Here on the Psorophora species, you can kind of see those like Circe or those last things present that might be a little bit more receded in depending on your sample quality, but you'll still see this kind of V-like shape rather than this rounded shape that you see on the left right here. So if our abdomen is bluntly rounded, we know that and all those other steps before, we're thinking of our Culex species. Whereas if our abdomen is sharply pointed, we're going to be thinking of either Aedes or Psorophora. And then, so if we wanted to differentiate between our Aedes and our Psorophora, the next thing we're going to look at still on the abdomen is going to be looking at this banding pattern that is on these abdominal segments. So Eric went into this before, but as a reminder, this is going to be very, very important throughout for genus level identification, but also with your species within species. So we're going to be looking whether these bands are basal, so coming from the top of the segment, as we see here. So in red, this is just outlining one abdominal segment right here. So you can see these white silver scales are coming from the top of that segment, so basally before. Whereas if we look at this one on the right here, it's a little bit more difficult to see from the dorsal view, but from the lateral view, you can see that these bands are apical. So they're coming from the bottom of that abdominal segment or after, right? So again, as a reminder, right, if we have a pointed abdomen that has basal banding, we're going to be thinking of our Aedes species, which we're going to be going into depth today. And if we have a pointed abdomen with apical banding, we are going to be thinking of our Psorophora species. And like I said before, right, this is really important right now, but this basal versus apical banding pattern is not only for the abdomen, it's also used on the legs. So it's going to be very important throughout your identification for various different genera.
And it doesn't have to be just basal or apical. It can be both. You can have basal and apical banding on the same mosquito.
Yep. So just as we're about to go over just a review of these species, but in general, these are just shared characteristics of each genus. However, like Eric said before, there's always exceptions to the rules. You know, there's thousands of different mosquito species, so there's always something that's going to be thrown in there. So it's always important to just be to understand what's locally happening in your region and what species you locally have to understand those exceptions to rules. So first, right, we're our Toxorhynchides. So these are some typical common features. So we don't like to use size a lot when we're talking about ID, just because a lot of different things can affect mosquito sizes. The amount of predators in the water, the amount of nutrients in that water when they're in their larval stage. A lot of environmental factors can all play a role in how big your mosquito is. But Toxorhynchides is going to be always a very, very large mosquito. If you have one in your petri dish when you're doing your ID, it's going to be just obvious before you even look through your scope that you're going to see these mosquitoes sticking out right here. As we said before, the defining feature that helped us get to this is their strongly curved proboscis. And they're also going to have covered with intensely colored iridescent scales throughout the mosquito body. These are a really great mosquito. So sometimes they're called the mosquito assassin or the elephant mosquito.
Cannibal mosquito.
These are really great because as adults, the females do not need blood meals. They do not take blood meals. They do not need that to produce their eggs. And as in the larval stage, they predate on other mosquito larvae. So these are a really great biological control agent that we have been raising in our lab here.
Probably should mention the curve in the proboscis with Toxorhynchides is really, really evident. And sometimes you'll get other specimens after they die, the proboscis will kind of have a bend in it. But it's a major difference between a slight bend post-mortem and this strong curve here.
So next, we're going to talk about our Anopheles. So again, the major thing here, if we know that we have a female mosquito, we've gone through we know our sample's female, that their palpi are the same length as the proboscis. And when we look at then to speciate these, they're going to have a majority of them are going to have very discernible scale patterns on the wings. So that's going to be the best place to look. And we're going to be going over our Anopheles in depth later today. So we're going to be really focusing in on the wings and the scale patterns on those wings to differentiate the different species. Next, we have our really cool Uranotaenia. So these are the major thing that we talked about here is that the thorax, but also the wings and the abdomen are going to have patches with these iridescent blue or white scales, but these are very, very obvious. Again, like with size that we don't really love, but Uranotaenia especially are extremely small. You may sometimes think that there's some sort of bycatch or non-mosquitoes when you look at your petri dish, but be on the lookout for them. They're extremely small. They have these really shiny blue scales that are going to tip you off and be glittering in your scope. So, and then another feature that you can look at with these to help us identify them is at the end of their proboscis, they're gonna be a little bulbous at the tip. So it just becomes a little bit more wide at the tip of that proboscis.
I will say that Uranotaenia is one of those species that often has a curve in the proboscis, but there's honestly no way you can mix up Toxorhynchides with Uranotaenia just because of that size difference.
There's a paper I got to find somewhere that was published a long time ago. I don't even remember who published this, but they did an overwinter survey with this genus, which includes Lowii and Sapphirina, at Fort Jackson, which is in Lower Plaquemines Parish. And they went in this fort and they had hundreds and thousands of Uranotaenia overwintering as adults in there. And for a species that doesn't take blood meals, they were, I guess, opportunistic enough that this guy was actually receiving bites from Uranotaenia. Super odd. But typically, no, these guys won't take blood meals from people.
It's usually amphibians. Right?
Amphibians, earthworms, annelids, all that kind of stuff.
Cool. So next, the next two we're going to go over. First, we're going to go with Mansonia. So these are pretty large mosquitoes. The important feature that we went over is their broad asymmetrical wing scales. So again, the jasmine rice.
Jasmine rice.
Or how I like to call it, the oak leaves.
It depends on the species of oak.
I don't know the difference in trees. Wing scales are mixed dark and pale, which give them that kind of salt and pepper look. They're going to have that blunt abdomen, and then a very good defining feature for Mansonia versus the next genus that we'll go over is that on their hind tibia they do not have a pale band, but they have pale bands throughout the other abdominal segments, but the tibia itself does not have a pale band. Which in comparison, when we look at our Coquiletidia, so in the United States, we only have one species, Coquiletidia perturbans, but these are very similar to Mansonia, medium to large body with broad asymmetrical wing scales and a blunt abdomen. Again, they kind of have that salt and peppery look because of the dark and pale wing scales that are broad. But when we look at that, the hind tibia, near the distal end, they're going to have a white band on it, which is a very defining feature to differentiate that Coquiletidia perturbans with our Mansonia species.
And that band on the proboscis for Coquiletidia is pretty evident.
It's super wide.
Right. And
Normally
where
you can tell confined to the base.
if we look at Mansonia and you look at the keys for Mansonia, it's going to say that they have this band, but I'm not sold on that for identification. Coquiletidia will definitely have a band on the proboscis, a wide one.
Cool. So next we have our Culex species. So these mosquitoes are typically all medium-sized brown mosquitoes, kind of very difficult in the adult stage to really see the differences between them, but don't worry, we are going to do a really in-depth dive in our session 2 on our Culex species in the Gulf South. They're going to have a blunt abdomen, and on their abdomen, throughout the species, their discernible scale patterns on the abdomen are going to help us identify them in that species level. Culex are going to have narrow wing scales, and then the important thing right here from our dichotomous key and review is that subcostal vein CT is absent. So they do not have those armpit hairs, which when we compare it to our Culecida, and we can see this a little bit better right here, those CT on the underside of the wing are going to be present, which give it this really nice like armpit hair look when you're seeing this specimen from afar. Our Culecida are going to be very similar otherwise to our Culex, that they're brownish in color. Some are much larger than others, so that will help us also kind of see that something is different. And they're going to have narrow wing scales as well. Cool. And then finally, we'll move to our Aedes. So our Aedes, most are typically the dark bodied with silvery or gold scales throughout the body. Our Aedes are going to have our pointed abdomen, right? Narrow wing scales. And then when we're looking at that abdomen, we're looking at those basal band patterns on the abdomen. Versus when we are looking at our Psorophora right here, these are going to be large mosquitoes, similar with Aedes. They're going to have a pointed abdomen. But instead of that basal banding on the abdomen, they're going to have apical abdominal banding. Another feature of our Psorophora species is that they're going to have scale patterns on the discernible scale patterns on the legs or hind tarsi, and their hind tibiae are going to have long erect scales, which you can kind of see in this picture here, but it'll be very, very evident when you're looking at your samples that those scales are really coming off the hind tibiae.
Even if you have a leg in your samples that you collect, it's really easy to tell if you have a Psorophora somewhere in there, even if the leg is not attached to the body.
Awesome. So we're going to have another quick quiz right here. So hopefully it's going to pop up for you guys, but we'll give you 30 seconds. If it doesn't pop up, just think about your answer. But just as a review, female mosquitoes of which genus have palpi as long as the proboscis?
And as mentioned before, if you don't have it coming up, you get an A. Hint.
Aedes.
Yeah.
All right. Hopefully everyone could get their answer in. So the answer to this question is going to be our Anopheles species, right? So that's that main defining feature for our female mosquitoes. If their palpi are as long as the proboscis, right, it's going to automatically eliminate all those other genera, and we can focus in on our Anopheles. If they're shorter, then we can move to our next steps in our dichotomous key or in our genus level identification. All right, so just to kind of reiterate something that we've been kind of hinting at throughout this is that you're going to have differences in your sample quality, right? And that's going to be based off of a lot of different factors. The trap that you use is going to be a major thing, right? Like, for example, like a BG Sentinel trap where the mosquito is not going through a fan. They may preserve a lot more scales, or if you're using an aspirator out in the field versus if you're using a trap where the mosquito has to go through that fan, like a CDC light trap, which can really knock off a lot of those scales. Additionally, you know, how crowded that net is, the other bycatch that gets in there and, you know, feasts on those mosquito samples, how long it's been in the field, how you preserve them, all play a role in the scales that you're going to be able to seed on your mosquitoes. So here we have just a picture of the same, both Culex quincunfasciatus, but you can see how beat up this sample on the right is. Some scales are going to rub off easier than others, and we're going to in our next and our remaining slides, we're going to talk about. There's a lot of things that are used to help differentiate these species, but those scales are sometimes the first things that fall off. So it's really important to be comfortable with multiple different features to speciate samples because those scales may not be present on your sample to be able to do that pretty easy speciation.
And look, that's a pretty good beat up specimen too, because you'd be amazed what happens to these mosquitoes in the traps. He's talking about bycatch. I will be pulling mosquitoes out of the abdomens of scarab beetles, and so you've got nothing left. And this is where it's like okay to ID the genus, and don't stress about getting the species. Culex especially. You know, if you've got that Eraticus pilosis picator group and it gets beat up, you're not getting past that subgroup there.
Exactly. So yeah, so if there are multiple features that can help differentiate when you're trying to get to that species level, you be comfortable with all those and use all of those to kind of reiterate or recheck depending on the sample quality. And then, you know, if you were to have a beat up sample, like I believe that, you know, mosquito identification is not really about, you know, memorization of these things, or it's not like formulas or things. It's really kind of an artistic skill, right? Where look where those few scales that you have are remaining and try to make out those patterns. So like here, for example, if you have these scales that are like at the edge, let's imagine all these other scales are gone, right? And you only have like one or two scales in each little segment right here. You can start to kind of make out that shape and it can kind of help you get to that. But like James said, if you don't feel comfortable with it, do not force an ID, right? If you can get to the genus level alone, that's still very, very, very good.
And you need to also bounce off other resources, you know, not just restricted to your inner department, but reach out to other people. Like, for example, Mike Rowles is on this. You know, he's a great guy in Florida. He's a top taxonomist. Reach out to your other contacts, you know, and it's okay to ask other people for advice or for help on IDs, you know.
The other thing, like, why would species even be important for this, which we didn't touch on? We will in a little bit, but when we're talking about different vectors of certain diseases, some we're not concerned with at all. But if you're forcing an ID to species of, say, something that could be a potential vector of something and it's not, then you're just wasting resources on whatever comes after that, whether you're trying to treat for something or not.
So we're going to move into our Aedes species identification. So this kind of to reiterate Eric's point here. So Aedes are going to lay their eggs singly in a variety of different habitats. So getting to that species level can be very important in understanding the larval habitats of within this genus, right? Some species are going to be using man-made containers like waste tires and buckets, but also natural containers like tree holes, bromeliads, bamboo, etc. While others are going to be laying their eggs in temporal pools, edges of the soil. So even though getting to that genus level can give you a lot of information, that species level is really going to help you get a lot more information about the behaviors of those mosquitoes, so you can make that targeted abatement decisions to really help reduce your populations if needed. Our Aedes genus are going to overwinter as eggs, so they're desiccation resistant. They'll need that drying stage, which is a lot of the times why, like especially the ones that are laying their eggs in these floodwater areas, come out in such massive numbers all at once. But our shared characteristics, again, to reiterate, are Aedes. They're going to have pointed abdomens, right? And then they're going to have this basal scaling pattern on their abdomen.
I know we probably didn't go over this in this talk because it's more about identification, but just to mention how much water does Aedes need to actually lay eggs. Like it could be as little as a bottle cap that's laying around. So when we're talking about a control, you have to look for every source of water that they could possibly be breeding in.
Leaf axils. No, there's some plants like in the genus Tradescantia, the spiderworts. Those leaf axils collect a lot of water, and they can have Aedes breed in that.
Yep.
I'm breeding a whole bunch of Aedes right now.
So just as a note, so the following slides are not going to be inclusive of all Aedes species found in the Gulf South, but the subset that we've kind of included here are species that are commonly found in our region, as well as species that share many characteristics and may be misidentified with each other. So again, we're not covering all Aedes species, but hopefully if these are ones that in your area, it'll help be like a little review or helpful for you. And if they're not in your area, they may one day will be there. So it's good information to understand these species that may not be in your region currently. Again, as always, important to familiarize yourself with all species in and around your region, so you can be prepared and not be taken, by surprise when you're doing your identification.
One approach I like to take when it comes to doing mosquito ID, because you'll have 100 mosquitoes dumped in front of you. Where do you start? And it's very overwhelming if you're at a beginning level. I first time I was getting ready to do ID, I just got in my car and left. But what I like to do is, if you're running these gravid traps and we're targeting mostly Culex fasciatus, you would kind of just go into that knowing that, so you're going to be expecting that, and just look for other things. Look for other things that stand out, and look at the different ones.
So, just in general, the ones that we're going to be going over, of course, our main ones, which are Aegypti, albopictus, and vexans. We have a little bit of distribution, which I think we have range maps on that. But the reason we're really concerned about these is their medical importance. Things like Zika, dengue, chikungunya, West Nile virus, dog heartworm, they're kind of all over the place with what we're looking for.
And so this is just to review and for you guys' knowledge, if you want to re-watch the recording, but just as an overview really of the medical importance, like Eric's saying, and why we care about them. So it's very important to understand maybe like obviously their distribution, their larval habitat, kind of like their flight range, especially too, but also their medical importance.
And as I said before, the reason why species does come into play, as you can see with Aedes informatus, of medical importance, there's none. But homeowners will say, hey, we're being torn up by these mosquitoes. Are we concerned about vectors? No, but the homeowner's concerned because they can't sit in their yard and things like that. So that's why we have these up here.
Awesome. So the way we structured this is we're going to go through go over various species, and we're kind of comparing and contrasting these ones that look alike. If there's no really good comparison in our region, they'll just be standalone. But for the beginning of each section, it will have kind of these comparisons. So we're first going to start off with Aedes aegypti versus Aedes albopictus.
All right, I'm not sure where everyone's from, but Aedes aegypti has been introduced to a lot of places, so you've probably got it unless you're further up north. They will fly and bite at any time. They tend to be a little more crepuscular, but I can tell you, just sitting in my yard, it really doesn't matter. They'll lay their eggs on the side of walls of containers, and some examples of that, crawfish trays where someone I live with keeps plants. They'll lay the eggs on the side of that. Fountains, bird baths, that kind of thing. And they use a skip over position behavior, which means they'll lay a cluster of eggs here and move over, wherever the opportunity is to lay those eggs.
But
They'll
not
skip
all in
over
the
that.
same spot.
Right.
Which makes the control very important, right? Like if you have neighbors that have, various different buckets and they knock over one, that's not good. You're still going to have a problem with these. They're not just going to put their whole batch of eggs into one water source. Another important thing, just like as we talk about getting to that species level, like I like to highlight here, especially with some of the species that are coming up, our Aedes aegypti are very weak flyers. So they're only going to go around like 1 to 2 blocks pretty much from where they emerge, which is very important for our control. If we are looking at our traps and we just get to that Aedes species, we have we say, okay, we have Aedes in this trap. We have 50 Aedes, right? Okay, well, then they could have a flight range of, 1 block to 5 miles, 10 miles, right? So how do we do control? Whereas if we can get to this species level and we know, okay, we're capturing these mosquitoes, there must be a larval habitat within this home, this block, or these surrounding blocks that we can target and really treat these sources.
So looking at the morphological features here for Aedes aegypti, I'm going to start off by looking at the scutum. It's got a really nicely pronounced lyre shape there, kind of like a harp. And again, depending on the condition, it's usually pretty well pronounced. That's what you're going to start with there.
Yeah, and again, like James says, like this is a very evident one of when it's perfect and great, you're going to have a really easy time seeing this very discernible lyre shape, right? And when we're talking about lyre, it's a harp, so there's the outsides of the harp and then the strings. But this, unfortunately, this scutum scaling falls off pretty quickly or gets very beat up very quickly. So it's hard to discern if it's that harp shape. So I know a lot of people go to this as the only thing to differentiate, but we're going to show some other things that will help you differentiate this mosquito.
So we'll pretend that harp shape is completely gone. What do you want to look at next?
You have to pretend that harp shape is usually gone.
So the next feature that I personally like to go to is the clypeus here, which is the shield right in front of the face. Those scales, again, will usually be there to some degree. They'll rub off too. But in aegypti, it's going to be silvery, kind of white colored scales, and there'll usually be some left on there, if not the whole thing. The palpi are tipped and white, but that's not a separating feature between Albopictus, but for other species, keep that in mind. Dark scales on the proboscis, so there's no bands. Keep that in mind, no bands at all on this animal.
Now, my first part that I go to, I skip over the key, and I always go to the underside of the abdomen. And if we look at the underside of this abdomen, it's going to be mostly white. A lot of times this will be desiccated a little bit, but you can always see that where that white patch of scaling is underneath the abdomen.
So 3 features right off the bat that we can get to aegypti. The legs will have banding on tarsomeres, black and white.
Basal banding.
Very similar to aegypti is Albopictus, and I suspect most of you are familiar with this. Color scheme is very similar, except it doesn't have that lyre shape. It'll have like a racing stripe down the center of the scutum. Aggressive biters is just really kind of putting it mildly, man. Come sit at my house. Again, very similar to aegypti in its mannerisms. Container breeders, cups, tires, kids' toys, leaf axles, cat litter containers that are sitting outside.
James's backyard.
It's not just the backyard, it's the kitchen too. Again, weak flyers, just a few blocks away, and it is a competent vector of West Nile, Eastern equine encephalitis. We don't have to worry about California here, but also lacrosse, chikungunya, dengue, and Zika viruses. So we do pool these two if we have enough specimens from each trap. A better shot of the scutum here showing that racing stripe. It's pretty well pronounced. It will fall off too. So let's pretend this is not there. What did we look at before with the Egypti? Raise your hands.
Nope.
I guarantee you someone raised their hand.
I did.
Again, here with Albopictus, like Egypti, will have no bands on the proboscis. So let's look at the clypeus if that scutum stripe isn't there. Egypti had silvery scales on the clypeus. Albopictus will be all dark.
But they're going to share, like Egypti, they're going to share that their palpi have silvery scales at the tips. And like James said before, the proboscis is dark, but this main differentiation right here is when you look at the clypeus.
Eric's favorite.
And as I said, I never start there because those scales fall off always. But if you look at the underside of the abdomen of Albopictus, it's going to have these spotchy scales instead of that broad white scaling underneath. And this, I mean, I think 99.9% of the time that is there.
And there's no right way to start. Like it is whatever you want to do with it. Eric finds that the easiest. That's what he started with. You know, someone else go to the clypeus. It's just there's no really wrong or right way to do it. Whatever you can put in your mind to remember it. The legs with elbows are going to be very similar to Egypti. The banding.
Basal
That
banding
leg's
on the legs.
broken, but you know.
Yeah.
Yeah, the bands.
So just to review some of the similarities between the two and why people can really easily misidentify them with each other, right? They're both these black and white mosquitoes with no banding on the proboscis and white scales on the tips of the palpi. And they both are going to have basal banding on their legs. So each one of these segments is coming from that closer before, right? Basal banding from each abdominal segment. I mean, sorry, from each tergite. Not tergite, sorry, I'm so sorry. Tarsimer. But when we look at our key differences, right? So there's three major ones that we covered, right? So like James said and Eric said, you can choose whichever way to start, but it's always good to have multiple different ways to kind of confirm what you're looking at. So the first, we can look at our scutum with that lyre shape for Egypti versus that racing stripe shape for Albopictus. We can look at those abdominal sternites. So from underneath, if it's solid white or segmented, so these basal bands of pale scales on the sternites. And then when we look at that clypeus, for our Aedes aegypti, we're going to have our white scales in patches. And like we've been talking about, they may be knocked off, but when you look and focus in there, you may see a few remaining ones if you're seeing those white silver scales versus completely dark scaled on the clypeus.
One thing I do want to mention with Egypti, using the word variability again, on the scutum, sometimes the CD will have a really gold tint to it. And for those that are familiar with Aedes japonicus, you know, you could almost have a start, you know, when you see that gold. And I think Mike Riles is going to touch on japonicus in his invasive talk. But be mindful that some of these Egypti can have more gold scales, or CT rather, on the scutum than others.
Cool. So that was our, easy one off the bat, but now we're going to start moving into a little bit more difficult ones, but this one's great. Aedes infirmatus versus Aedes atlanticus.
So this little group here is in the subgenus Ochliotatus, and they include the floodwater Aedes. So we'll touch on infirmatus and atlanticus here. As the name indicates, temporal pools. So you get a heavy downpour where there wasn't water last week, and now you got Aedes laying eggs here. They are day biters. And this here, infirmatus, is the most common of our woodland Aedes, like we term it. This particular species is not known currently to transmit any human pathogens. And this was first described as science from Baton Rouge, 1906. You remember that? So looking at the morphological features here for infirmatus, if I had a penny for each time that these things were in good shape, I'd have a penny. But these are good specimens here, and you can see that scutum pattern is very broad. It's usually silver or goldish, and it is not complete. It stops at the scutellum back there. So you've got a good shape specimen. It's pretty cut and dry here when you're comparing it with the next species, but it just that stripe ends. It doesn't continue all the way through.
They never look like that, by the
I'm
way.
telling you, you're having a good day when you get one like this. So just a closer shot of that scutum stripe.
And the important thing too, you know, when we're talking about, you know, these kind of terms like broad and narrow, which can be somewhat subjective. And if you haven't seen these species in a while, it's hard to kind of compare what is broad compared to the next when we're looking at Atlanticus next. But what you can see here is that the light colored scales are going to be broader than the lateral dark scales. So just comparing the size of the width right here versus the size of those lateral scales in the scutum, you're going to see that this white scaling right here is broader, and that's what gives us that name, the silverback mosquito, right? Because it has this nice broad silverback when it is present.
There are a bunch of silverback mosquitoes, though.
Yeah. Thorax is pretty unremarkable. You know, there's a grouping of scales there that are probably going to be similar to all the other ones that we're going to follow. The antepronotum and the post-spherical area not have pale scales. Abdomen, yeah, pretty much just dark scale dorsally with white scales laterally, kind of forming a triangular shape there. They don't cross over the segments whatsoever, carrying, so they won't form a full band going across the top of the abdomen. And kind of back up, talking about the broad versus thin. If you are familiar with a lot of these mosquitoes and maybe not Infirmatus, you can compare the width of the stripe to, is it broader than an Albopictus? If you're familiar with Albopictus already, oh, it looks broader than that. So you can also bounce it off with other mosquitoes that you're familiar with. Whatever you can do to store it into your mind, you know, a feature, whatever works for you that reminds you of something, go with it. Some people like to say, oh, it looks like a saw blade or a sawtooth. I mean, whatever you need to put in your mind to memorize these features is best for you. Legs, unremarkable, no banding. They're all dark. And Atlanticus is a very similar species. And we don't see as many Atlanticus as we do Infirmatus. It's another floodwater breeder, and their peak season coincides with hurricane season. And I had really never seen Atlanticus in numbers till Hurricane Ida. Hurricane Ida. And there were all these Aedes on my side door, you know, and I was like, oh, right, I got a new species for my yard list. And I went out there, I was collecting Atlanticus. I had collected about 200 just on the side, and in the side yard. I couldn't even make it to the backyard, man. I wouldn't be sitting here right now. So this is a primary vector for keystone virus, which is a subtype of California encephalitis. So generally indistinguishable from Tormentor, and we'll kind of not even touch on Tormentor unless Mike Riles gets to that. They are best distinguished as larvae, but if you have pristine shaped specimens, there is a slight fork at the back of the stripe, and we'll look at the stripe in the next slide here. This is a complete stripe, and it's gonna go all the way back from the front to the back. And in Tormentor, this section here will often have a fork, and I'm just going to leave it at that because we don't see enough of both of them, and I don't want to confuse anybody with Tormentor. But that stripe is narrow. You can see the side, the dark scales on the side are broader than the stripe itself.
I'll just go ahead and say it again. They never look like that.
That one on the right, though.
I know.
That's just a closer shot. You see there's rub there. You can see where there's some missing, that long grain rice. I'm hungry too. The thorax is almost identical to Aedes infirmatus. In fact, I don't know if there are any differences. That's just a lateral view there. The triangles again.
So very similar to Infirmatus.
Yep. And the legs, same thing. No banding on the legs, so we're going to have all dark legs with Atlanticus and Infirmatus. Now, again, depending on the condition of your specimen, this may or may not be of any use whatsoever. But Atlanticus will have the prominent, like triangular shapes, dark scales there behind the occiput. And that's a differentiating factor between Atlanticus tormentor and Dupreei, which is a tiny thing. And we won't even really talk about Dupreei.
So kind of for this, these two comparison, these are very, very similar mosquitoes in a lot of ways. They both have lateral patches of white scales at the base of each segment. The abdominal terga are dark scaled dorsally, and their legs are dark scaled with no banding. And this is a very important fact, right? Eric has mentioned it, right? Like the scutums are not going to look pristine like this many times, but this is kind of where like looking for the remaining scales is extremely important. If you focus in and you are seeing white scales on these edges that make it broader, that's going to help you differentiate your Aedes infirmatus from your Atlanticus. So let's say, for example, we're looking at this, and it's really, really beat up, this picture on the right. If you are seeing some white scales like further out, that would make that section is the light scales are broader than the lateral dark scale sections, then that's a really helpful way to kind of get your way closer to Aedes infirmatus rather than Atlanticus, and vice versa. If you're not seeing any of those scales there, that may help you differentiate the two. So it's really important, even if you don't have perfect pristine samples with this, to look for where those remaining patches lie on your scutum. But again, Aedes infirmatus will have that broad stripe versus Atlanticus will have that narrow stripe. And also, look at the end of that. Look at the end right here. So if it is an incomplete stripe, you're going to feel more confident with Aedes infirmatus. Whereas if you see those white scales at the end, a complete stripe, that will help you differentiate that and make the determination of that it's Atlanticus. So just another quiz for you guys. Should pop up in your box. So the question is, a shared characteristic of Aedes species is? We'll give you 30 seconds.
You're asking me or the class?
I'm asking the class, not you.
I was asking me. I wasn't paying attention.
That's right. I was playing hangman.
All right. So we'll give you another couple seconds. So the answer here is going to be D. It's going to be a pointed abdomen with a basal scaling pattern on the abdomen. So reminder, that's what's going to be shared amongst all of our Aedes species, especially the ones that we're covering today. Close answer choice here would be A, pointed abdomen with apical scaling pattern on the abdomen. That would get you to that Psorophora. So next is Aedes vexans versus Aedes canadensis. Take it away, James.
Aedes vexans is our most common floodwater mosquito. And they'll breed on neutral grounds, in your backyard, the front yard, anywhere you get a heavy rain downpour, temporal pools. This is going to be your guy and gal. It's going to be one of the floodwaters that has banded legs. So the two that we just covered, infirmatus and Atlanticus, do not have banded legs. These do. It says painful bite. I mean, what mosquito do you want to be bit by? But I mean, painful bite should be accentuated with these guys because they are absolutely relentless, and they can fly over 10 miles, and probably one of the most common competent vectors of canine heartworm. The scutum has gold flecks, CD on top, which again are usually there, but this one here is kind of halfway beat up and missing a bunch of his eyes too.
Yeah, and it's not very remarkable scaling pattern like we've seen on the other 4 that we've just gone over, where they had these real shapes, like the lyre shape, the racing stripe, the broad scales, right? This is just kind of unremarkable gold speckled scaling on the scutum. Yeah,
you're not really going to use it to separate anything. The palpi and the proboscis are going to be dark scaled, so there's no bands there. Thorax has got them rice grains on the side. Now, let's see what we're going to call this. I'm going to go with jasmine rice on those too. But they're always going to have those clusters there on the sides of the thorax. Scutellum with the gold flecks, which we had already mentioned. It'll grow. So, all right, looking at the abdominal pattern here, picture this as an inverted V, if that'll help you remember vexans.
We have a picture on the next slide with it.
Yeah. And most of these are in decent enough shape to where you can kind of see that.
So what James is showing here, right, is looking at this little notch that's on every abdominal segment. Imagine this little V in there. And when we look at it at the next picture where we just have it shown in, just a very easy way, and it works out really great. V for Vexans. So a very easy, like mnemonic or kind of way to just remember that when you have your Vexans, you're looking for that inverted V notch on your abdominal segments.
I should also mention that those scutellum scales that are listed as gold can be variable. That can be silverish as well.
Even the abdomen can be variable on these.
Yeah. Oh, yeah. Absolutely. And then there's subspecies, that
Some will have like a median stripe down the middle, but that's a different species that we're going to go over that with.
as I mentioned before, the basal banding on the legs, and they're typically going to be more narrow than some of the other Aedes. And the other one with banded legs that's close to it is Canadensis. We don't see a whole lot. And I think if we collected maybe 20 of these since I've been here, that's plenty. But it seems like there's little pocket populations here and there, and they're really heavily forested species. And looking at them really quick, you could just pass that off as a Vexans, and especially some of those golder colored Vexans. So the palpi and the proboscis are also dark scaled. That's not going to help you. Like Vexans, it has patches of light colored scales on the thorax, so that's not going to help you.
Well, that one's not going to help you.
And this is the condition of this one, certainly won't, but just to show, the patches of the light colored scales do cross over laterally on the dorsal side of the abdomen.
Yeah, just the important part here, this is not a great picture because it's pretty rare for us, but they do not have that inverted V notch. So that is going to be one of the main defining characteristics from your Aedes vexans, because everything else makes it pretty resemble Aedes vexans.
Which were subspecies become vexans.
But there is a really easy way to identify Canadensis from Vexans. If we look at the hind tarsomeres, they're going to have both basal and apical banding, which I said before. As you can see right here on this top one, you can see that the banding is on both sides of that segment right there.
Yeah, so this red line here is denoting the end of that segment. So you can see, right, that you have apical and basal banding here. And then, do you want to talk about Mathesoni?
No.
Yeah, it's kind of unremarkable, but there's the two subspecies of Canadensis, and Mathesoni has dark scales on the final tarsomere, which we don't have a photo of because we don't have any specimens of that subspecies.
I think we have one.
Do we?
Mm-hmm. But
similarities, they're very similar. The legs are going to be your best bet there because both species have dark palpi and proboscis. The brown coloration with the light colored thoracic scales. So if you're going to have one that looks like this, just go to the banding on the legs. If your abdomen's in good shape, you can use the V shape there. But oftentimes the shape of the V's is different, and it's not even a subspecies thing. It's just one of those individual species that can have some variability.
Oh, sorry. Did you want to say something about that?
no, I'm good. So Aedes solistans versus Aedes tanuricus. So solistans, probably my favorite mosquito here. Found all along the Gulf Coast. They're strong flyers. They can get up to 40 miles with the right winds, but usually its average is about 5 to 10 mile range. Solistans is one that we get quite a few calls about whenever they're out and about.
Especially in March. People that live out in Venetian Isles, they just get slaughtered by solicitans.
Yep. So tan and yellow scales covering the scutum, and this is actually whenever you see them, it really stands out compared to.
Oh, sorry. Sorry.
Whoa, where are you going?
Sorry.
All right, so it really stands out when we look at them in the lateral view, where the gold is on the top, where this white scaling is going to be on the sides. They do have this broad band on the proboscis. That's really important to know that it is a broad band on the proboscis. The palpi have white tips, as you can see, but man, once you see that band right there, that's what you're dealing with. As mentioned before, they have these white scalings on the side with the gold scales on the top.
And they'll be covered.
Yeah, those contrast really well. And they, this is a species that does hold a pattern pretty well, probably because of the amount of scaling on it.
Right.
That those clusters of the white thoracic scales stand really well. And they contrast super well with that mohawk on top
And
and that band.
so they as well have basal bands, but they'll have this longitudinal stripe. This stripe wears off very often. I don't go by it. I mean, if you have pristine specimens, it'll be there, but as mentioned, it's usually the thorax that we look at.
Reiterate though.
So
So again, just to reiterate, when we're talking about this longitudinal stripe, just because it's the first time we're seeing a scaling pattern like this, right? Again, they have these basal bands in red, or denoted by these red lines. And then if you look in this green box, this longitudinal stripe throughout all the abdominal segments can be present on Aedes solicitans.
and sometimes that can be present on Vexans as well. It's there. We talked about this before. I can't remember what it was. They have dark legs with white basal bands, but on the first tarsal segment, they'll have this mid white band in there. There's a couple other species that have that as well that we'll go over next week as well. You can see it. This one's a little faded, but you can tell right there. That's what we're looking at. The one that it gets confused with most often somehow is the black salt marsh mosquito, or Aedes taeniorhynchus. These are usually going to be darker colored, and you can see that it doesn't have that contrast on the side of the thorax right there. Actually, we've had quite a few of these this past week as well. If you look at the band on the proboscis, it's not that broad band like it is in solicitans, right?
That's correct.
So it's a narrow band right here on that proboscis. They also have the dark palpi with white tips, as you can see in this picture on the left.
And again, when we talk about that narrow versus broad, right? If you spent all winter, not catching anything, your first ones of the year, when you're looking at that broad versus narrow, it may be difficult to start. But if you're starting to have both of them at the same time in your region and your traps, then that broad versus narrow will kind of become more evident to you. But again, it's very subjective of what is narrow when you have nothing to compare narrow to.
It's not only subjective to that, but also wear too,
Right.
So it's really great if you're going to start out with these two to have specimens of both and just look at them side by side.
That's what I was just going to mention. So we keep a voucher collection here for the city of New Orleans, and it's always good to have pin specimens, just so if you do find something, you can always go back to your vouchers to compare and contrast what you have. So as mentioned before, the scutum is going to be dark brown scaled with narrow golden yellow, as you can see right here.
It doesn't contrast as much as it does in solicitans.
Right, you can obviously see on the lateral view right here, it does not have as much white scaling as solicitans will. Of course, the white basal bands are present on the abdomen. It doesn't have that dorsal stripe as in solicitans would have. Dark legs with basal white bands. And then we compare similarities right here. Oh, yeah, if you go back. So it doesn't have that white band in the middle of that segment right there as well. Similarities, well, they're similar, but not really. Right here you can see, let's go to the differences.
Okay.
So key differences, as we usually say, that lateral stripe right here, a longitudinal stripe on the abdomen, as with no longitudinal stripe on taeniorhynchus.
Oh, go back one. I do want to point out the dots there on the dorsal side of taeniorhynchus on the right are not typical. Some individuals will have them and they'll be absent in others. So that's not really a feature that I would look for to separate between taeniorhynchus and its solicitans.
And differences, as mentioned before. So broad band right here on the proboscis of solicitans, as with narrow band right here on the proboscis of taeniorhynchus. And then I don't really think many people confuse these two. Once you see them both, you can, especially if they're together in a trap, you'll know the difference.
If you get a good look at solicitans, you can actually, aside separating it from Aedes mitchellii, which we're not going to cover, you You can tell it right off.
You don't even need a scope.
Yeah.
If you have a good one. As mentioned before, that white band present in the middle of the first tarsal segment, whereas it's absent in Taeniorhynchus, is another difference. Aedes triceriatus, anybody?
Don't matter. Yeah. Aedes triceriatus is the eastern treehole mosquito, and there's a sister species, Aedes hendersoni. Just for the sake of this, we'll say they're identical. They need to be separated by larvae, and in some cases, they can be separated by adults. I've seen some specimens in Ohio with Eric, and then Evan that works here photographed a really nice hendersoni from Alabama, I believe. But otherwise, these two species look identical. They are black and white Aedes like the albo types, but you'll notice that these guys don't have banding on the legs. They are a vector of lacrosse encephalitis and others. And like the name indicates, they will utilize tree holes, but other man-made stuff too, Mardi Gras cups,
Tires.
that kind of thing. It is a day biter. So the scutum, looking from the top view, it looks like someone took an airbrush and kind of just put these two little white stripes down the side, like it's got eyebrows on it. The center of the scutum is all going to be dark scaled. So if it looks like it's got those, I call them a saddle of white scaling on the sides, even though you're dealing with triceriatus.
This is another species that you don't really need a scope to identify with this part.
Yep.
With the white scales on the side of the thorax.
Yeah, it's just so pronounced. And then the scaling behind the occiput too is really white and pronounced. The abdomen has got dark and white scales. They're unbanded dorsally. That white femur there.
I will bring this up about the abdomen too. I know James mentioned triangular shaped a lot for Informatus.
Yeah.
You can see this is not going to be triangular shaped at all.
Additionally, triceriatus, a lot of times when they're in good shape, will have bluish tints. They're not blue scales like Uranotaenia, but they will sheen blue, or more so than some of the other Aedes. The proboscis and palpi are dark, so they're pretty, unremarkable. Scales are curved and erect right before the occiput here, which you're really going to need some high-powered magnification to get to. But your specimens in good shape, they stand out like a mohawk.
Yes.
Legs, like we mentioned, they're all dark, so there's no banding. Immediately you're not dealing with an Egypti or an albo type. And I mentioned that white femur earlier too, right there.
I think Mike is going to talk a little bit more about that.
Yeah, Mike's got some good information on that.
All right, so I think this might be our last Aedes. I think
This
so.
is the hardest one.
This is the worst one to identify, actually. So Aedes fulvus pallens. We don't see very many of them. They're another woodland species that we have. I'm just gonna ask you guys to tell me in the comments what stands out about this one besides other Aedes, right? So they're large mosquitoes and they're yellow and black in coloration. So
Slam dunk.
there you go. Done. Just joking. So the scutum is yellow with two large black spots depending on which subspecies you're talking about. So this one's Fulvus pallens, so they will have these two black spots on the scutum. This is a terrible picture of this one's not yellow, but it should be.
Yeah.
Just imagine it's yellow. The palpi and proboscis are yellow and they have these black tips on them. Some people call that banding. I don't call that banding. I just say it's a yellow proboscis with a black tip on the end. The legs also have these black apical bands on them. You're not going to confuse this with anything else. I'm not even going to compare it to anything else, but we will do a brief overview of different ones. So if we're talking about banding on the proboscis, we're really talking about Aedes solicitans and Aedes taeniorhynchus. All of the other ones will not have a band, so including Fulvus pallens. So if you see a band, you automatically go to those two.
And again, this is pertaining to the species we covered today, right? But
Yes.
but this is a good way, we think, to like understand your local species, and you can break them apart and make some educational materials for your lab, for your people, just to have these as reminders. And it really helps speed up that ID. as you practice, that genus level is going to come automatic, right? So if you're still stuck on that genus level identification, that's totally fine. It's going to take time, but the more you practice, then that will start becoming automatic. Then once you get to the species level, instead of having to key out all the ways and go through all steps, you can think about certain features or certain banding to kind of help you filter out what you're thinking about. So like this is an example, and we'll have another slide, the next one. But if you like for the species that we covered today, if we look at our proboscis first thing and we see banding, we can eliminate all of these things on the right-hand side because they do not present with that banding pattern.
Now, if something is slightly different, just know that it can be something that we didn't know was in our area before. So you might have to go to other keys as well. These are just the common ones that we have in New Orleans. Banding on the legs, you can break it down that way, right? No banding would be Infirmatus atlantis tricereatus, whereas the other ones in our area will have banding on the legs. The scutum, there you go.
Yeah, so with Aedes, for a lot of the species that we covered, the scaling on the scutum can be very defining, right? So again, just another way to kind of think about your overall species that you have in your region and to kind of break them apart. That will help teach new people that are coming in your agency to learn these IDs. It's to like look at these certain characteristics and compare and contrast them so that you can look at all of them in general. But again, there's always going to be ones that don't have remarkable scaling on certain things, like these ones in the bottom where the scutum scaling is not alone going to help you kind of differentiate them. So we're going to have another quick quiz because I know we want to get to Anopheles.
But we're only going to cover 4 Anopheles, so we're good.
Yeah. So quick quiz. What is one defining scaling pattern that can differentiate Aedes sollicitans from Aedes taeniorhynchus? I'll give you. So we'll give you like a couple of seconds to get your answer in. All right. So the answer here is going to be that longitudinal stripe present on the abdomen. These other answer choices, they both share these characteristics. For this answer choice D right here, which is kind of to throw you off, right? That band may be different looking in terms of size, but they both have it present. So to differentiate Aedes sollicitans from Aedes taeniorhynchus, that longitudinal stripe is one of the various different ways that we went over that can help you differentiate them from each other. Cool. So next we're going to move into Anopheles identifications. Like Eric said, we're going to go over 4 major species that we find in the Gulf South. Just as an overview for Anopheles, these are going to be permanent water breeders that lay their eggs singly with floats. Their larvae are going to be laying parallel to the water surface. They do not have that siphon, so very easy way to, not only from the adults with those palpi that are as long as the proboscis, but in larvae, very easy way to identify them or to get them to the genus level by looking at they do not have that siphon. So they lay parallel on the water surface, which is also really cool to see in comparison. Just as an overview here, we have, a photo of both Anopheles males versus the females. So that easy way to differentiate them here is that you look at those antennae for that male, very, very bushy. And when we talk about bushy, like look how different it looks, like a paintbrush, like especially a used paintbrush versus the female where it's a lot less. And then even when you look at the palps, these palps are a lot more bushy than when you look at our females. And if you were to be able to look at the last abdominal segments, you would be able to see those claspers or the cerci on those male samples.
So wing veins, as I mentioned before, there's a few that are really important for Anopheles, but we'll go into that whenever we see them. This is just an overview. So Anopheles crucians versus Anopheles bradleyi. We've actually been seeing a lot more bradleyi lately. These are, as with all Anopheles, they're permanent water mosquitoes, very common across Southeastern United States. It's a complex, so this exists as a group of closely related organisms. So when you're going through the keys for Anopheles, sometimes it can be confusing. Crucians, first thing we look at, well, we know it's Anopheles, we already mentioned that, but the palpi are going to have 3 pale bands on them, as you can see where the red arrows are pointing. And the proboscis is dark. The thing with these palpi and the pale bands, these can easily be rubbed off. So a lot of times we have to go somewhere else, but this is usually the first thing that we look at to see where we're getting.
And for the ones that we're going to be comparing to, even if you just have like 2 of them remaining, it'll be helpful
Yeah,
because we have 3 sets of these pale bands on the proboscis, on the palpi, whereas some of the other ones that we're going to go over have no scales on the
And
palpi.
you
So
can, I mean, yeah. And then the other thing, yeah, with rub, you have to be concerned with that. So sometimes if you have an all dark palp, it can look like it has bands on it because those CD have rubbed off. So you just have to zoom in further to make sure that's not what you're looking at.
the thorax is does not have any pale scales, otherwise pretty unremarkable.
So wing venation, or not venation, I'm sorry, wing scales are really important when we're looking at Anopheles. These aren't always there either. When we're talking about crucians, we look at that distal end of the wing where the red circle is, and there's going to be white scaling right there. As compared to other species, there might be other spots besides that one, but this is really important with crucians. As you can see closer up, that little white pattern right there will get you to crucians. So with knee spots, these are not going to have knee spots, what we call knee spots, on the end of the femora right there. So these are going to be all dark scaled legs. Bradley eye.
So Bradley eye is very similar to crucians, and when I mean very similar, like you've really got to take your time with this. Let's just say they're morphologically identical. The larvae are different, but there is one key feature which we'll show you in a second. They prefer brackish water. So this is what I'm thinking, why we're seeing more Bradley eye in some areas than others. Places like Virgil, places like Sankofa, they have more stagnant water than around here. We're finding Anopheles Bradley eye in decent numbers in only a few spots that seem to be consistent. One trap will have 130 crucians and nothing in terms of Bradley eye. But then I did a trap yesterday that had 110 Bradley eye and 270 crucians. So the common denominator is that stagnant water. I think Bradley eye prefers that over crucians.
You can see on the palps here as well. As I said, with crucians, they will have those 3 bands of white scales.
And here's a good example of one where that third one where the laser pointer is over, it's not very evident, right? But you still have these other 2 pale bands that can kind of help. So that third one kind of rubbed off on this sample. Just like crucians, right? The thorax is going to be without any pale scales, otherwise unremarkable.
So James really likes wing venation, so I'll let him take this one.
Yeah, man. So you're saying we've got an Anopheles here with banded palpi, and the thorax is the same, the abdomen is the same. How are we going to separate this from crucians? Because it also has that cream-colored tip to the wing. Well, the answer is very small, but if we go to the CU vein right here where Alex is hovering, you'll notice those scales there are kind of gold. They're going to be gold or white, always in Bradley eye. In crucians, the scales on that vein will be either entirely dark or mixed white and dark. But if you've got that CU vein right there that is entirely light scale, then that's a separating feature for Bradley eye here in this case.
Oh, they like to rub off too.
Yep. Yeah. Again, so what do you do? Well, you just can't, sometimes that's all there is to it.
So for this first slide, we're just showing you what would be shown in a dichotomous key, just reiterating what James said. For crucians, this is going to be entirely dark scaled here or a mixture of dark and light scales, whereas for Bradley eye, it's going to be entirely light scaled. And then when we look at live photos right here, you can see that similar thing. So here on our crucians, we have a mixture of those dark and light scales, which I think have animated right there. Great.
Perfect.
Whereas in our Bradley eye, when we look right here, entirely light scaled.
Yeah, and you've got to look at it most of the time. If you look at it dorsally, it'd be better.
Yeah, but the thing that we talked about before, they're going to be shared with this pale spot, which is not shown in this Bradley eye picture right here, but they're going to have that tipped, white tipped at the distal end or distal end. Yeah. So our next two that we're going to go over are Anopheles quadrimaculatus and Anopheles punctopenis. Take it away, Vince.
Most commonly known one for malaria transmission is quadrimaculatus, although several species in the complex are also vectors. Permanent floodwater species, swamps, marshes, rice fields. I don't have this breeding in my yard.
You like rice?
Yeah,
Okay.
yeah. The palpi does not have bands, but now pay close attention to all your Anopheles because for some reason the scales on them palpi will fall off, and you'll be like, okay, well this thing has bands. It must be a Crucians complex, but it's not. It's just missing scales. So the palpi here will not have any bands, and then the vertex will have those stiff, pale CD kind of forming like a mohawk behind the head there. Like every other Anopheles, it really does not have much in the way of thoracic scales. And we'll go back to the wing venation here. On the photo in the bottom left, you can see the 4 clusters of dark scales. Pay no mind to the white ones. So that's where the name comes from, quad. There's grouping of the 4 scales. You'll notice the distal end of the wing does not have a cream-colored tip, and the cubital vein is all dark scaled. Legs.
All right.
Yeah.
This is subjective to me. So as opposed to Crucians, they will say quads have this light knee spot. I don't use that for distinguishing often. Usually there's enough on the wings to go by that, or if you look at the palps.
Really, I think the knee spot thing is usually used more separating quadrimaculatus and Atropus.
Atropus, yeah. So when you're separating quads and Crucians, it's usually pretty easy.
Yeah, and like they said before, right, the palpi can be a defining feature, but if those are rubbed off, really the wings are your gold standard. And looking at the banding pattern, if you have those 4 black boxes for quad 4, just an easy way to remember, that's what you're looking for on their wings.
Punctopenis is one that really kind of stands out in terms of Anopheles. This one and Pseudopunctopenis, which we don't see. But this one here, the wing pattern, which we'll get to in the next slide, just like all the other Anopheles, they feed on mammals, including people. They're crepuscular. I imagine they'll bite during the day too. They are vectors of malaria in the southeastern U.S. And like other Anopheles, they'll breed in pools, puddles, ponds, artificial containers, and so forth.
Let's look at the side of the thorax.
Oh.
Yeah.
So first we'll start with that.
Like everything else.
Like every other Anopheles, there's no remarkable thoracic scales, all just kind of chocolate. And then we're looking at the palps there. It looks like they might have bands, huh? You see that? That's what I'm talking about. When those scales fall off, it can look like bands. That's when you want to zoom in all the way, as close as you can get, to make sure that it's rub and not light colored scales. The wings. What's different here? Don't everybody answer at once.
I'll answer.
All right, what you got?
I don't know.
All right, cool. So look at the top of the wing here. You'll notice all the other Anopheles that we looked at previously, it does not have that little section there that's pale. That's going to separate it immediately from the Crucians and the quad types.
I don't think I've ever seen a punctopenis that was missing.
No, that's always there.
But it also has that white scaling at the distal edge of the wing. But it's obvious whenever you see it. So let's look at them all.
Yep, that's a good comparison right there. You'll notice the Crucians, the top left cubital vein.
Sorry.
Yeah, right there. It's got light and dark scales. The distal tip is cream colored. Moving on to brightly eyed, that cubital vein is light scaled. Shares the pale spot at the distal end with Crucians and Punctopenis, but Punctopenis has that top row there of light scales. Quad's missing all of it.
Quad just wants to be itself.
Yeah. And the reason we put quads and Punctopenis together, right, is that Punctopenis, and depending on the sample you have, they may have some pretty remarkably like dark and light wings compared to some of these other Anopheles.
Mm-hmm.
So you may automatically see these dark spots in Punctopenis and be like, oh, I have a quad, but really focus on this patch of light scales up here, which quad will not have. It'll be completely dark scaled. Cool. And then if we look at our banding on the palpi, just as a review, reminder that when we look at our crucians and our Bradleyi, we're going to have palpi with 3 white bands, and our punctipennus and quad dramaculatus are going to have entirely dark scaled palpi. So thank you guys so much for being with us for these 2 hours. I know that was an information overload, but we're really excited to have put this on for you guys. We're going to be, next week, we will be going into Culex, Psorophora, Uranotaenia, and some more. And so please join us next week. But if there's any questions, we'd love to answer them. And just as a reminder, if you're interested in joining our Mosquito Academy, where we go through these species and hopefully some more, you'll have live specimens to really harken these identification skills and see these up close and not just look at pictures, but really have samples that you get to beat up and look at all different angles of. So please join us in September for that class as well. But if there's any questions, we'd love to answer them now, or you can email us. Please use the top 2 emails and not mine.
No, wait a second. Use the middle one.
All right, so
email address.
guys, amazing, right? So these guys are like a round of applause for all of our taxonomists. They're awesome, right? So super great. We've been monitoring the chat. Look at how awesome it is. Thank you, everybody. We have people from all over the world. I'm so excited. Please email education@nola.gov, right, with the information that we needed in order to get you your certificate. We've been taking attendance. We were paying attention. Don't worry about the poll, because I know many of you just put the answers inside the chat. So we're great there. Look, we're going to use the same link, and I've been emailing multiple times. So maybe many of you probably received the link again from while they were speaking, which is great. So again, really just make sure that you're checking your email, and if you feel free to share to your friends and colleagues. I mean, the whole goal here is for us to get this message out. So I don't see too many questions. I mean, there is a lot of questions about getting the presentation, but what we are going to do is to post this right onto the YouTube channel. So I think that's going to be super important. And look, I would absolutely sign up at vectoroutreach.org. We have resources there for all of you, and we are loading more and more every single week. So everything from social media content that you can download and just put your logo on it. We are working on presentations. We actually have quite a few ready to go. They just need to be formatted for Gulf South Vector, and then those will be PowerPoints that you can download and use for training as well. So again, thank you, everybody.
Claudia?
Yes, you saw some questions?
Yes, I do see some questions. The first one
cool.
is, how do Aedes cause dengue?
I mean, yeah, that's a great question. So, I mean, just different viruses are like different genera and different species are competent for different viruses. So, for a mosquito to be able to transmit a disease, that virus needs to not only enter through the body, go through the midgut, escape the midgut, and get to the salivary glands, but be able to replicate within that mosquito. So, different species are more competent than others to be able to transmit diseases. And then within, even within those species, you have different capacities within that, just based on different regions of the world, right? There's many different, even like an Aedes aegypti here might not be as competent as an Aedes aegypti somewhere else in the world. So that's why not all species of mosquitoes can transmit these diseases. A great example is Anopheles, can have the plasmodium of malaria, like go through that whole process, that intrinsic incubation period, and survive, whereas that's not going to happen with these other species. So different species are competent for different, or different species have different competencies as vectors. Hopefully that answers their question.
It is, and I just put it in the chat as well. This is Claudia again. We have a lot of PowerPoints presentations that are in there, and we have some basic information on mosquitoes, which does include many of the viruses and bacteria, not bacteria, and parasites. So please make sure you go check it out. I think you'll be able to get a lot of information there. Stacy, did you see any more questions?
Yes, when you talked about the 29,000 mosquitoes in the trap, which trap was that? What type of trap?
Yeah, that was
a CDC light trap.
Yeah, that was a CDC light trap. Obviously, different traps are going to target different species or have more narrow or broad species that it kind of tracks. So that was a CDC light trap in one of our more like rural areas, marshy areas. And it was after, lots of drought. And then we had a big rainstorm, put it out a couple weeks later, and that's when we had an explosion. I think in that one was Nigripalpus or Sal's in that. But those CDC light traps are really, really good of getting that wide range of or a lot of diversity. Whereas some other traps that we utilize, like our gravid trap, which we utilize a specific lure that's very attractive for Culex fasciatus, but not so attractive for other things like Aedes species, Psorophora, etc.
I will say when you get a trap that's like 1,000 mosquitoes, you let out a sigh, but you really let out a big sigh when you get a trap that has 29,000.
I sighed a lot of times when I was like
29 times.
Okay,
really questioning my life choices.
someone asked about Aedes pertinax.
Pertinax.
Yeah, Aedes pertinax. That's
Pertinax. Sorry.
yeah, that's an introduced species that we've not seen here yet, but Mike Riles has lots of experience with it in Florida.
So I think that might be covered in our session 3. Mike is really, our session 3 is going to be a lot about is really about invasive species. So some are going to overlap with our first 2 sessions, but also he's going to go really in depth on these invasive species, which you will go over that. So make sure you join session 3 because he has a lot of experience with that and some really great slides and pictures of how you can speciate that a sample.
Yeah, if you go to Google Scholar and search for Aedes pertinax, Mike Riles, you can pull up at least one of his publications that he's written up on it.
Okay. Oh, go ahead.
Yeah, actually somebody asked about, so how do you count 29,000 mosquitoes, guys?
Oh, geez. Well, over the course of some time, those clickers that you bought me are very nice. I'll say that. Yeah. So once the goal here is once we reach, for example, that trap, I would separate 300 per species to be tested. Anything out, after that is lanyard, but to get the whole picture, you count them in clusters. If you have this trap had predominantly Culex salinarius and Nigripalpus. You put a group of 5 to 10 together, you look at them, they're all the same thing. You count them by 5s and 10s if that's the majority of what you've got.
Yeah, I will say just the 2 things to add to that too is, James and Eric have been doing this for many, many years, and they're extremely, they're very, very good at what they do. So they're able to kind of quickly get to that species level without having to go through a key for all these, for these specimens of things that we see all the time. Some agencies, if they have a situation like that, they may subsample where they will, weigh out the mosquitoes. They'll take a portion of that, identify that certain amount, and then can estimate what the amounts are. In other cases, it really just depends on your program and what you want to get from your information. Even just from a bird's eye view, if I see a trap that has that much, it's a lot of info. Once James tells me what's the kind of main species, I can already start acting on that until that can be fully identified. So it really depends on what you're really wanting to do with your surveillance and your timeline of getting it done. That can be something that, can sit in the freezer for a while to really get all the way to the end, but at least we get the important information of, yes, this area of our city has 20,000 mosquitoes in one night. Okay, we need to do something about it.
I find the easiest way to count 29,000 mosquitoes is to put that trap back in the freezer and make sure James gets it. You just have to hide it underneath traps that are like lower than that number. All right.
Someone asked if you could elaborate on, I think this is during Anopheles, the fanzi.
Oh,
Oh, so Stephen's eye. Yeah, we don't
Yeah.
have
Stephen's eye.
Stephen's eye, so I don't know anything about Stephen's eye.
The CDC has a lot of information on that. We had, I mean, we have specimens here, but they're voucher specimens from the CDC. So they have a lot of information on their website for that.
And I will say, I'll reiterate, this is we're building our identification documents. That's ongoing. So that's something that we hope to add into our educational materials, and hopefully we'll have by our Mosquito Academy, and working with other agencies to kind of get samples that we don't currently get in New Orleans. If we just trap here, we're kind of limited to having great specimens and pictures of the things that we have around here. So we're building this program out to include more and more species down the line. So stick with us, and we'll definitely try to be adding a little bit more to these species that we go over in the coming events.
Okay. As a vector control staff, do you recommend that we use the same chemical using all the same chemical at the same time?
So I'm a little bit confused by that question. But let me see if I can understand. So I would say, it's a very important part of mosquito control is understanding the resistance in your local populations. So doing things from CDC bottle bioassays and KDR testing to also doing cage trials. I would say follow your label. But we utilize, like, for example, for adulticides, we have different chemical classes that we utilize. But obviously, like, one truck or one helicopter is just using one product. We're not mixing products at all. That's never the case. But we are rotating chemical classes, but we also may use multiple ones, but we're looking at our resistance profile. If we have high resistance to pyrethroids, we're not going to be using pyrethroids. So just it's a very important part of your program is to understand your localized or your population's resistance profile to make those decisions of what products that you do end up using.
But if you're using things for like mechanical, like Cocoa Bear or whatever, you're not gonna get.
Yes, I mean there's
like that or BTI.
There can be resistance to some larvicides, right? But that's why, again, utilizing multiple different classes, rotating periodically is always the best practice, so that you do not, and also, being mindful of when you do use these chemicals, right? Don't overuse these products when not necessary. I'm talking about adulticide right now. Is utilize it as a tool for vector control, but it's not the end all be all, right? It is a great way to knock down and break the disease cycle, but your population level control may be best used at larvicide usage or public education or source reduction. So utilizing all different areas of the IPM pillars is going to be a really great way to not create that resistance in your populations.
Okay. Have you seen morphological changes in mosquito species due to crossbreeding, climate change, or pollution in any area in the United States?
Actually, I think, not, I don't know on that question, but that kind of reminds me of what we saw with the Aedes vexans and the Aedes
sows with the spots.
So if you want to talk about like that one that we've been noticing with our Aedes vexans populations.
So some variations are more consistent than others. You're not going to have people throw around the word hybridization quite often, and it's just not the case. But so one example, Aedes vexans, will go back to that inverted V shape on the dorsal side of the abdomen. Some populations here, Texas, Maryland, maybe, and a few other states are, including Florida, are finding vexans with the white longitudinal dashes going up the dorsal side of the abdomen, which is morphologically consistent with the subspecies Aedes vexans nipponiae from Korea. We initially did DNA on one sample, and it kind of came back vexans, but it wasn't cut and dry. So now we're sending all of our samples to some folks in Florida. They're going to do DNA to determine whether this is the Korean subspecies or if it's just a one-off. But so many around us have this, and I wouldn't even say 1 out of 1,000. It's really not that common.
I mean, there's variation. I don't know whether you can say it's that due to climate change or anything. I mean, they're just
No.
different specimens, just like people are different, right? We're all the same species, but different. So you will see a little variation,
Yeah, and
but
that's
what
not.
it's due to is who knows.
And I think that's like to reiterate that point. We, like a lot of we avoided using coloration a lot of times in this talk because even just based off of where those larvae are, like where those larvae are developing and what is around them, you may get adults that look very different. We've had quinks that are green. We've had quinks that are red, so like that can be these like variations that may just be the environment that is in. But yes, I think that there are variations within these species that are important to look at to see if it's a new species or a new subspecies that's coming into your population, or like Eric said, there's just kind of general variation throughout your mosquito populations.
Okay. Claudia is answering some questions in the chat, but one that didn't get answered is, did we release any of the SIT mosquitoes here in New Orleans?
No, we have not been doing SIT. I think it's a really cool potential control option that we may look into in the future. We did attempt to research or attempt to look at releasing Toxorhynchides mosquitoes. So you didn't have to do the SIT process, but they can predate on some other species that are going to be laying in those like tree holes and things like that. So we have been exploring different biological control options here. We utilize also, mosquito fish and copepods as well. But we haven't really explored the SIT or Wolbachia or any of those mass release things. But I've seen a lot of the Florida districts have been doing that, and they've seen a lot of success. Obviously, they are, a temporary solution, but they can be a really cool other option rather than just relying on adulticides alone or larvicides alone in your program. So definitely something to look into. And as it continues to be something that gets adopted throughout the United States, I'm interested to see that, and maybe we will add that to our program in the future.
Okay. I think that's all of the questions.
All right. Well, guys, thanks everyone that's still on the line. Thank you so much for attending today, and we look forward to seeing you next Friday. If you have any issues with registration, please make sure you reach out and we will take care of it. All right. Have a great day. Thanks again. Thanks to our speakers. Bye-bye.